I will now continue with the second talk of the symposium about non-invasive neuromodulation of deep brain structures. In particular, in this project, we investigated the modulation of the human striatal activity by means of the temporal interference stimulation, the technique explained during the first talk of the symposium. To be highlighted, this is, as the previous case, one of the first studies applying TI in humans. In the current study, the technique was applied in the context of motor learning. Why? Well, because we know that motor learning is a fundamental domain we use in our everyday life, from learning how to use a tool to learning how to play an instrument, for example. Furthermore, population is now aging, and age comes with a lowering in motor performances and with an increased incidence of neurological and neurodegenerative diseases. An example is stroke. Here you can see how the incidence of stroke increases with age, and after the accident, a great number of patients is left with motor impairments. These are just some of the reasons why motor learning has already been highly characterized and studied. Several neuroimaging studies revealed a complex network underlying motor learning processes. In this complex network, the interplay of both cortical and subcortical regions is actually fundamental. Here you can see, for example, several of the areas implicated in the motor learning functions. And in between them, the striatum is one of the main hubs, located more in depth in the brain. Because of its position, this structure has mainly been investigated via animal work or in a pure associative way via neuroimaging studies in humans. What we know is that the striatum can be subdivided into three main parts, based on the functions and on the connections with different cortical areas. From the more anterior to more posterior part, we can find a limbic, an associative, and a sensory motor part. These subunits have been shown to differentially be implicated in different parts of the motor learning process. This process is characterized by an initial fast online learning, followed by a slower learning, consolidation, and long-term retention. Already in this first online fast learning, striatal activity showed a shift from associative regions, including the caudate, more involved in the initial part of the motor skills acquisition, to sensory motor regions, including the putamen, more implicated later in time. So, we now know that we want to target the striatum in the context of motor learning. As a next step, it is important to define which kind of protocol it can be used to modulate striatal activity and how. So to reply to the first question, the choice was made to use a well-established protocol mainly used in the TMS domain, the theta burst stimulation. Theta burst stimulation, TBS, demonstrated the ability of inducing brain plasticity, and in particular, one of the protocols originally introduced in humans showing plasticity modulation is the intermittent theta burst stimulation. In this specific case, trains of 2 seconds of stimulation are delivered, followed by 8 seconds of break. Each train is composed of 10 bursts at 5 Hz, the theta component, and each burst is in turn composed of 3 pulses at 100 Hz. To answer the how question, we then exploited the novel temporal interference stimulation able to go more in depth than the current non-invasive brain stimulation techniques. By combining 2 high-frequency currents oscillating at 2 kHz, we hence created 2 stimulations, a control and an active one. In the control stimulation, which we will define as high-frequency control, no shift was applied in between the 2 high-frequency currents, leading to a flat envelope which would hence not act on the neural membrane. In the active condition, instead, a shift in frequency was introduced to mimic the intermittent theta burst pattern with the resulting envelope. The ability of this technique to maximize the envelope more in depth allows to, for the first time, study behavioral causal relationship between the striatum and the motor learning process in humans. Based on this information, our main hypothesis of the study was then that ITBS-TI stimulation would be able to reach and modulate the ongoing task-induced brain activity within the striatum and, by doing so, to modulate behavioral outcomes in humans. So, to recatch a little bit your attention, I will already give you a little bit of insights in our main findings. What we found was that ITBS-TI stimulation led to, firstly, a differential pattern of local brain activations within the striatum, our target region. We then observed an increased bold activity within the motor network, the network including the striatum, during the motor learning task. And finally, these brain changes also reflected in an increased motor learning task performance on the behavioral level. I will now guide you through each of these points to explain you more in details how we arrived to these conclusions, starting from, firstly, the differential pattern of local brain activations within the striatum. In order to do so, what we did was to ask 15 young healthy subjects to undergo an MRI study with a randomized, double-blinded crossover design. We firstly performed 2 resting state MRI sessions with at least 3 days in between them, in order to investigate possible effects of the stimulation at rest. Participants performed 3 scans at each visit of around 8 minutes before, during, and after stimulation, and either the active or the control protocol was used in a randomized order. Data were pre-processed in order to reduce the artifacts effects which could have been introduced by the setup and especially the stimulation. And after that, a seed-based connectivity analysis was performed with the right striatum as seed, and no significant difference was found between ITBS and HF control. This suggests that, at least at rest, TI stimulation did not induce any observable brain changes. In the second part of the study, we asked the subjects to perform 2 task-based fMRI sessions. During the acquisition, participants had to perform a motor learning task, namely the sequential finger tapping task, with their non-dominant left hand. This was chosen since it is an established task in the field, able to well study motor learning curves. The task consists in reproducing a sequence of numbers shown on the screen with a 4-button box, with each finger associated to a specific number from 2 to 5. The sequence was shown during the whole duration of the task in order to reduce a working memory load, and the subjects were instructed to perform the task as fast and as accurate as possible. Each session consisted in 30 seconds baseline block with no stimulation and no images acquired, and then this was followed by 6 blocks of around 9 minutes and a half, each composed of 10 30 seconds repetitions of the motor learning task, intermingled with 30 seconds of rest. And during this resting period, a fixation cross was shown. Our main question was then to look whether the stimulation was affecting striatal activity. Thus, as a first step, we extracted the average bold activity within the striatal region of interest As a second step, we were interested in looking at the dynamics within the striatum. As in previous literature, it was shown that there is a shift from associative to sensory motor part of the striatum, from more the superior part to the most inferior part. We then looked during our task at which subparts were showing either a linear increase or a decrease in activity with time. Here you can see the different parts of the striatum depicted with different shadows of green, and in hot color scale you will see areas showing a linear increase of activity with time, and in cold colors there are regions with decreased activity in time. In line with previous results, an increase was shown in the inferior part, whilst a decrease was observable in the superior part of both stimulations. To then better investigate the dynamical changes along time, we also plotted the evolution of the peak activation for each of the stimulation protocol. In this case, a shift from the superior to the inferior striatum was more visible during the ITBS stimulations, whilst the location was more constant during the control. These qualitative results suggest that ITBS stimulation can support the striatal shift already observed during the motor learning process in previous studies. Okay, so up to here we could see a specific topographic modulation of striatal activity. Then, as second main finding, if you remember, we also found an increased activity within the whole motor network. How we did this? We started by looking at the whole brain activation during the two conditions, active and control. Here I show you the control condition, and you can nicely see the activation that is mainly focused in the motor network regions, which showed significant higher activation with respect to baseline. This is a confirmation that our task successfully engaged the motor network, and it also supports the good quality of the data with, for instance, primary motor areas on the contralateral side more activated. Then, when comparing the whole brain activation between the two sessions, you can here see that a significant higher activation was induced by the ITBS condition in the motor-related regions, including the striatum 2. You can in fact see here clusters in the cerebellum, thalamus, the supplementary motor cortex, and all of these regions are related to motor learning. Finally, we observed that the changes at the brain level were also reflected in a change in the behavior, with increased motor performances during the motor learning task. But now I will describe you more in details how we arrived to this conclusion. To specifically study behavioral effects, we performed an additional study outside the MRI environment, again with the sequential finger tapping task. In this case, subjects underwent a shorter protocol of around 20 minutes with longer blocks of 90 seconds. After a first baseline block, again without stimulation to assess initial performances and correct the results for inter-subject variability, subjects performed 7 blocks of 19 seconds of the motor learning task, intermingled with 90 seconds of rest. In the behavioral results I am about to show you, the main outcome is represented by the number of correct sequences performed during each block, corrected by baseline. When looking at the results of 15 young subjects, here you can see that no difference was found between the two stimulations. We then investigated the same protocol in an older cohort, which represents a suboptimal model of the motor network. In this case, ITBS stimulation led to higher motor performances, up to 33% of improvements with respect to the control. These results, the difference between the young and the older cohort, could be explained by different reasons. Firstly, the older cohort could simply have a greater room for improvements because of the decreased motor performances due to age. An alternative hypothesis instead could be the fact that age leads to both structural and functional neurodegeneration affecting brain plasticity. The stimulation could hence support brain plasticity, pushing the brain, let's say, toward a more younger dynamic. An Another factor which we then found to be important for the effects of the stimulation was the dose of both stimulation and training. During the longer protocol performed in the MRI scanner, in fact, subjects performed a 3 times longer protocol than outside the scanner. When comparing the normalized correct key presses, even in a younger cohort in which before we didn't observe any effects of stimulation, in this case the stimulation appeared to have an effect with ITBS leading to higher performances than with respect to control stimulation. This points towards a dose dependency with longer protocols able to make effects of the stimulation emerge even in the younger cohort. Let me now summarize the main findings again of the current project. In the study, we were able to demonstrate that intermittent diverse stimulation delivered with the temporal interference technique led to, firstly, a differential pattern of local brain activations within the striatum, our target region. In this case, we observed the average activity within the putamen and the caudate, subparts of the striatum, and we observed that already during the task, the putamen was showing a higher activation with respect to the caudate. And specifically in this region already activated by the task, we observed a modulation of the activity with ITBS leading to higher activation with respect to the control stimulation. This, together with the resting state results in which no difference was found in between the two stimulation protocols and at rest, these results point toward the necessity and the importance of a pre-activation of the target structures in order to observe the effects of the stimulation. We then also saw how the shift that was observed previously in literature from more associative to sensory motor part of the striatum was actually supported by the ITBS stimulation, which showed visually this shift, whilst during the control, the location of the peak was remaining constant. As second main results, we also observed an increased both activity within the motor network during the motor learning task. In fact, the motor network, which includes also the striatum, showed higher activity during ITBS with respect to control. And finally, these effects observed at the brain level, they also reflected into increased motor learning task performance, so behavioral effects. All these results taken together really point toward a good modulation of the striatal target by means of ITBS TI stimulation. And thank you very much to Professor Huma and the whole TI team, which allowed to put in place such a great project. Thank you.